And I would like to take some questions. The first question is from Mr. Bharat Das, Dr. Bharat Das. He's asking, what is the difference between diagnostic stewardship and laboratory stewardship? Is there any government organization which helps small health setups for different forms of stewardship measures? I think this question goes to Dr. Malini.
Yeah, Dr. Bharat, like when we talk of diagnostic stewardship, it means like we are in some criteria, clinical criteria, radiological criteria, the test or the microbiological or biochemistry, that criteria, and what are the treatment we are giving to the patient. So that's diagnostic stewardship. But when we talk of laboratory stewardship, there we have pre-analytical, analytical and post-analytical. For example, I'll give you an example. I'm a microbiologist. So diagnostic stewardship, if I'm talking of fungal infection, so there I'll have a clinical criteria of fungal infection, radiological criteria. For example, invasive aspergillosis. So tree in bud sign or halo appearance on CT scan. Then clinical criteria will be hemoptysis and other signs and symptoms. And then you have the microbiological criteria, that is galactomannan antigen testing, you have KOH, you have fungal culture. So that's the microbiome. So if I talk of diagnostic stewardship, so that's a broader area. Clinical, radiological test is also there, and the treatment, whether I have to give amphotericin B or voriconazole or a combination. So as part of that EORTC criteria. So that's a diagnostic stewardship. But when I talk of laboratory stewardship in invasive aspergillosis, galactomannan antigen test, pre-analytical, how's the sample, analytical and post-analytical, only that particular test and uska pre and that galactomannan antigen testing. So you got my point. Diagnostic stewardship is broader area, including the, clinician is also there, the test is also there, and then the treatment is also there. So that was my answer. Yeah.
So I will just add, can I add something, Dr. Malini, to this?
Yeah, sure. You can
Yes,
add
please
it, that pre-analytical and analytical and all.
No,
Yeah.
there was this second part of the question was any guidelines. So guidelines are GCLP guidelines of ICMR. Although they are applicable to the organization or to the labs who are doing clinical trials, right? But if you apply those guidelines, because they are based on ISO 15189. So if you apply those guidelines to your diagnostic center, to your small center, because patient sample is patient, so whether you are receiving one sample or 100 samples, you need to follow a certain protocol. So you can follow those guidelines. And in addition, on NABL portal also, this ISO 15189 is easily available in the form of NABL 217. So if you download that, you will see starting from pre-analytical to the post-analytical, what all is required. Because these guidelines are to safeguard yourself. If some medical legal case happens, you can show that these are the steps which you have followed, and you have documentary evidence of each and everything. I hope the answer is sufficient.
I think it's sufficient, madam. And I will take the another question, which is on how to safe dispose of the urine samples and vacutainers. I will add on that. Will you brief that how a sample in biochemistry and pathology, like EDTA or serum vial, how we dispose of these samples, including urine?
Yeah, the good practice would be, good practice would be the, like as per WHO, it is best available technique, best available practice. So I would like to ask Dr. Bharat, like you have a small volume laboratory or a large volume lab? Okay, I'll just answer. See, if you're in a peripheral area or a PHC lab, a very, out of bound areas, so there you are just getting 7-8 samples. So using an autoclave would be, not the right, you're consuming a lot of energy and all that. So in a peripheral setup, you can of course disinfect, and then its respective category, disinfect in a bucket and sieve model. Like if you have just 6 to 8 samples. So but then again, if you're in a large volume or you're in a lab, let's say a medical college or a district hospital or a small private lab, which has got, let's say more than 30 samples. So there you could have, more than 50 samples. There you could have a small autoclave. the pressure cooker kind of autoclave with the standards. But if your lab is big, you have, let's say you're in a hospital which is more than 1,000 bed. So there you could have a waste autoclave with HEPA filter. So that's my take on that. So it's pre-treatment. If the sample size is less than 10, you can do the bucket and sieve model there. And if your sample size is big, you can have a small autoclave with a, digital this thing. And then if your lab is located in a big hospital, you can have autoclave with HEPA filter. So anyway, you have to do pre-treatment, whether it is sodium hypochlorite, that is just if they are less number of samples, and otherwise it is pre-treatment by autoclaving, and then it's respective category. They are normally of plastic only, so it will go in red.
Thank you, Dr. Malini. I think the next question is also for you. Is there any guideline related to sample collection from pediatric patients, like to minimize panic while collecting?
Yes, CLSI GP41, that is collection of diagnostic, venous blood specimen from the pediatric patient, that like you have to use 23 gauge butterfly needle, then how you comfort the patient, how you counsel in front of their guardians or parents. So it's all mentioned there, and I'm sure, in the quality manual also it is there. Yeah, okay.
The next question is, any guidelines for monthly acceptable CV limit in clinical biochemistry? Goes for Dr. Mamta. Dr. Mamta is there. Can you little bit elaborate your question? Monthly CV of what? For the parameters, or?
Must be for parameters only. So monthly acceptable CV limit. So when whatever parameter we are doing, as you must have seen that there is some CV given in the kit insert, whatever reagent you are using, there is a kit insert for that. So first refer to that, what CV percentage they have given in that. So you start monitoring, because initially how do you monitor your QC? On the basis of the results values given in the kit insert. So on the manufacturer mean. So you start monitoring your performance. So after a certain, as per your policy, after 20 readings, 30 readings or 60 readings, you define your own mean and CV. Own mean and ST. So monthly you will have your CV. So once your CV is attained, so for a period of 6 months, you monitor your CV if you have same lot of control. So if you monitor your CV and then you see what is the maximum CV attained, not the average, maximum CV attained. Suppose in 6 months I have got 5, 5.9, 6.2, 7.1, and around 6.3. So 7.1 was the maximum. So that has become my limit now. So then I monitor my performance on basis of that. If it goes beyond that, then because it was the maximum, CV is what? Coefficient of variation. So that was the maximum variation which was acceptable. Then in addition to that, to your question, I will add, then there is a concept MU, measurement of uncertainty, because you started your CV process monitoring in 6 months. So there may be many factors which must have been variable. So if you there is a factor 1.98, if you multiply your CV by 1 point, which means it is approximately double. So if CV of any parameter like 7.1 is a benchmark, so if there is any variation or something, so up to certain level you can monitor. Beyond that, you need to take corrective action. And these are the benchmarks which are alarming, which indicates that you need to look into your QC performance if it is going beyond a certain limits. So that's why you need to have your limit.
The next question is also for Dr. Malini. How to safe disposal of mercury lamps can be done?
See, mercury is a hazardous chemical again. So this is again through authorized recyclers only. And if you have a spill from the mercury lamp, then you could collect them. There's a mercury spill protocol is there. You can collect the bead through droppers or through the cardboard cardboard sheets are there, and put them in a bottle which has got water, and the mercury pearls will just settle at the at the base, and this would await a recycler again. You can keep them in safe and secluded location and awaiting authorized recycler. One is this. Then the second thing is, if you have the extended producer responsibility, so best is, if you have hazardous chemicals in such, like, for example, in fluorescent microscope, you have mercury lamp. Then as per as, in the biosafety hood, as part of manometer also, mercury is there. So best is that these equipments, they are covered under extended producer responsibility. The manufacturer only takes them back.
The
And
next
otherwise,
question.
certain states are also declared mercury free now, like Delhi is mercury free, certain other states are also mercury free. So best is to store them in a safe location and awaiting authorized recycler. Again, okay. Thank you. And extended producer responsibility if it is there in the equipment.
Thank you. The next question is also with Dr. Mamta. Traceability of card test.
When we do anything by card test, so there are certain guidelines which you can we have CLIA guidelines, we have CLSI guidelines, and whatever kit insert you are using, again, I always recommend to everyone, whichever test you are doing, whether you are doing qualitative, quantitative, POCT, or any other semi-automatic or fully automated, just pick up the kit insert, read the kit insert, you will have all the traceabilities in that. Even like what we have for biochemistry, we have standard textbook teats. But you will notice that in each of the kit insert, the first reference is given as teats only. So because that has the strongest traceability. So whichever card method or whatever you are using, just refer to the kit insert and the guidelines, CLSI and CLIA guidelines.
Thank you, Dr. Mamta. The next question is also with Dr. Malini. How to dispose positive samples like HIV and HBsAg?
As per the universal precautions, all samples, whether they are positive or negative, they have to be treated as potentially infective. So the best method is again autoclaving and its respective categories. They are mostly these vacuotainers, they are plastic, 99.9%. So therefore, after autoclaving, that is pre-treatment, they will go to red category.
Another question is also with you, madam. How can microbiology PG students can contribute?
How can microbiology, see, when you've done your postgraduate at level, you get a 20 question on, waste management or this. When you're senior resident, you learn more about that. And when you're faculty, so most of the time you're taken in as quality expert or as biomedical based expert or Kayakalp. Again, that's a government program on biomedical based sanitation, infection control. So you're taken in the team as either the nodal officer or as one of the expert committee member. So that's the role of microbiologist in the antimicrobial stewardship, in the stewardship programs, in hospital infection control, in quality teams, in waste management, in Kayakalp. So as of now, as a PG, you need to dispose of, you need to segregate your waste and in the color-coded bins and bags and follow the guidelines and answer your theory paper, which you'll get around 20 marks on this, 20 marks on stewardship or hospital infection control or waste management or as part of quality in microbiology. But when you become a faculty, then you have to look after the practical aspect in the hospital as well as the lab.
The next question is from Dr. Guddi Hela. How we correlate laboratory finding with patient history? Dr. Mamta, would you like to answer that?
Sorry, I just missed the last sentence. What did you say?
How we correlate laboratory finding with the patient's history?
Patients?
History.
How we correlate?
Yeah.
There are, if you are part of a standalone diagnostic center or a standalone lab, then it is slightly difficult, but you can make it simpler. That is a kind of laboratory stewardship or diagnostic center stewardship, that whenever you're receiving any sample, take a small clinical history. said what is the reason that why this sample has been, because in TRF, in test acquisition form, whether again it is in a soft copy or a hard copy, just make a note of it, that what is the current state of the patient when patient is giving first time sample in your lab or in your hospital. So if you make a clinical note of it, if patient has any history, you can easily correlate it. And if there is any difficulty, just pick up the phone. If the number of the clinician is given, talk to the clinician. If clinician's number is not there, then talk to the patient. Suppose, for example, I can give you example. We received one sample, only one test, first time we got, and the glycated hemoglobin was 14.5. We repeated it. We didn't have any clinical history because the billing clerk had not entered any clinical details. So we had to call a patient because the clinician's number was not reachable. So we called the patient, and the patient very happily confessed that yes, he is highly diabetic. But then after that again, because there was a time limitation, any incidents which occurs, we need to educate our people that this is, and who will be benefited? You will be benefited, I will be benefited if we will have the clinical history with us in each and every test.
Madam, next question is also with you. POCT instrument need calibration and quality control?
Yes, they do need. Each POCT, like it depends on the manufacturers, the requirement, not even IQC, they require ECO also. These are the guidelines, and there are certain guidelines which are for POCT, and I'm sure all good institutes, big organizations are following that, because ultimately what happens, when do we need POCT? We don't use them regularly. We use them only in emergency. Most of the times we use diagnostic services. So if the values will not correlate, there will be a panic. So what happens when the lab, when you send sample to the fully functional automated lab or semi-automated lab, they have a system, device system. They calibrate their equipment, they calibrate their reagent, they run the controls and all. But POCT suddenly in an emergency will take the sample, just prick the sample and just put one drop on the sample plate. So in that case, there might be a problem. So that's why the NABH 6th standard has these guidelines that all the POCT should be under the lab people. They should be monitoring the functioning, the quality part, their reporting and all, so that there should be no confusion and no difference in the results, whether it is being done on POCT or being done on the main equipment. So it is very, very important. And as I said, guidelines. There are some equipment, like we are doing one particular test lead on electrochemistry equipment, lead analyzer. That particular equipment manufacturer has given us a certificate that this equipment does not require any calibration. So we are sure that our results are consistent and there is no malfunctioning or there is no problem with the equipment. But if the manufacturer recommends that 6 monthly calibration or something is required, then you must do it.
So I'll take the last question now. It's time is running out, and to conclude the
Just
session,
give me 5 minutes.
the
I'm
last
getting
question
back to
is
you.
last question is with Dr. Malini. What is the shelf life of autopsy samples and disposal methods?
It's a very good question, very rarely asked. You know, like autopsy samples, they have varying shelf lives depending upon the type of sample and the intended analysis. Like, for example, the tissue samples for histology, they are typically fixed in formalin. They can be stored for long term and 70% alcohol. But then samples for toxicological analysis, they are stored frozen. They are frozen and -20 at -20, -80. But you know, then how long they should be disposed of? Basically, it is the unopened specimen, they are often disposed of after 12 months. That's the timeline. And then there are, of course, there are certain disposal methods also, like they are tissues, so they'll go in yellow category. And if it is a infected plastic waste or infected samples, again, you will be autoclaving and then its respective category of red. And then there are certain general disposal guidelines also available. Then you have to follow the national and the specific state regulation also. Like, for example, you know, like in Mumbai, the autopsies are conducted, at night also. So, you know, that regulation and that kind of state regulation also you have to follow for the autopsy. Yeah.
I request participants not to send more questions. I got 2 more questions right now, and we are at the end of the session now. So whatever queries or you have, you can send it to us through email, we will, respond back. I'm taking last two questions. Which is better, BRI values, TEATS or KIT insert? Goes to Dr. Mamta.
Good question. If Dr. Harsh will permit me, I'll just want to give response to these questions also, which I can see. Two more questions, just briefly, lot-to-lot verification, BRI and this Westgard rules. So first we'll come to BRI. BRI, preferably we should take from kit insert, and we should verify those values from kit insert in our population by running 20 to 40 samples. And very soon ICMR guidelines are also coming for the BRIs in Indian population. So if you take TEATS, because those studies have been done on a particular equipment, if you go into the detail in TEATS, you will see they have written some specific, these BRIs have been established on Roche, Cobas or Abbott Alinity or something, these kind of equipment names they have given. So to be 100% sure, because you are using that particular kit insert, those who have done that validation and verification on particular number, many times 100 or 1000, so it is better to follow those BRIs, and specifically for immunoassays, because there is a lot of variability in immunoassay parameters. So personally, we follow always kit inserts unless we are sure that these are the kind of BRIs which are not correlating with our population. So majorly BRIs for that. Second question was, Dr. Harsh, can I take this one?
Yes, please.
Lot-to-lot verification, very easy guidelines, NABL 1128. These all are freely available on the NABL website. NABL, just go to NABL documents, search NABL 1128, you will see and download it. And just do Control F. You write lot-to-lot verification, you will get all the answers. You write LIS verification, you will get the answer. Lot-to-lot verification, preferably do with the patient samples and controls both, because kind of sample matrix you are using on your equipment. So it is more safer. Then Westgard rules, whether you are using one test or 100 tests, please use rules uniformly for each and every sample, whether you are doing on a small platform or on a big platform. Because what are Westgard rules? They are telling you that your QC is within the range. They are not outliers. And on basis of that, you are releasing your reports. So you just cannot ignore them. Okay. Thank you.
Thank you very much, both of the speakers, Dr. Malini and Dr. Mamta, for conducting session so well. I mean, their inputs are going to benefit all the participants, those who were present today and attended the whole webinar. So I would like to hand over to Dr. Mamta once again. Thank you very much, both of you.
Dr. Malini and
thank you, Dr. Mamta.
Heartfelt thanks to all of you, and I'll request Dr. Harshvardhan to please say a vote of thanks this time.
Thank you. First of all, I would like to thank Dr. Malini R. Kapoor for giving us a wonderful insight about beyond biomedical waste management, about telling, the latest guidelines and rules, how we can look for the good laboratory stewardship. And Dr. Mamta, was always very instrumental in organizing such events. And today also she proved it very right. That topic was really good, and I hope all participants must have enjoyed the session. And I would like to thank the KAHO team, which is the backbone of this webinar, and which has they are helping us from behind, Akshita and whole team of KAHO, which is behind all these successful webinars. And once again, thank all the participants. Dr. Ravi Kumar is also there, and whole KAHO team who is behind this webinar. Thank you very much. Thank you all.
So we are also thankful to Dr. Harsh to moderate this session beautifully. And I am also thankful to my organization, Sir Gangaram Hospital, from where I learned so much. And to by being an assessor also, so that quality is our forte. And also thank you. Any questions, please email to Akshita. We will get back to you. So thank you.
And the session will be session has been recorded, and it will be put it in the website, so you can view that. Thank you.
And special thanks to Dr. Malini, because whatever problem comes in waste, I always call her. So what we
Thank
do,
you,
how
Dr.
to do, Dr.
Mamta.
Malini.
Yeah, any questions, further question, I'll be there. You can share my email also, and we will collaborate in future, anything related to waste and stewardship. Yeah. It is a pleasure always with KAHO to, and the more such interactive sessions we have, we get to learn. And because, this autopsy question, this came up during COVID times. It was very pertinent. So I was very happy somebody asked this question. Yeah, good sessions and good interaction. Thank you, Kaho, Dr. Mamta, Dr. Harshvardhan Singh, Dr. Ravi Kumar, I can see, Dr. Ashok Ratan sir also, Dr. Aparna. Thank you, everybody. Yeah. And thank you. The participants were also very inquisitive and they were glued also. They were asking
Yeah.
pertinent questions. Yeah.
Yes.
Thank you. Yeah.
Thank you so much.
Thank you
Thank you,
so much.
Pramod, also from
Yeah.
Kao team.
Thank you, Pramod, and thank you, Akshita.
Thank you, ma'am.
Speechless connection.
Thank you,
Yeah.
ma'am.
Thank you,
Thank-
everyone.
Thank you, sir. Good night.
Good night.
Good night.